Good day, Prof. We are Group 1 and presenting on bovine viral diarrhea virus. My name is Pamela Basjan. So which possible samples would you take to send to the laboratory? If it's a live animal, then blood, abscess, joint, bladder, body cavity, organs, communicating body areas, and superficial body surfaces. And if it's a dead animal, then fluids, feces, urine, swabs, tissue, and blood.
Hi, my name is Samantha, and my question is, how would you preserve and transport these samples? The correct preservation and transport of samples is crucial to get accurate and valid laboratory results. One should preferably obtain specimens from live animals before any antimicrobial therapy has been administered, and samples taken from deceased animals should preferably be collected before putrefaction or autolytic changes can occur. It is important to minimize contamination when collecting specimens, and specimens should be stored in separate leak-proof containers that are marked with animal's identity, type of specimen, date of collection, and all other information that is necessary. In warm weather, it may be necessary to refrigerate samples. Viruses may also be frozen and transported over dry ice or transported on wet ice. However, this is often not necessary, depending on the virus that you are transporting. There are certain swab tube combinations that are both convenient to use and effective. Popular choices include sucrose-based and broth-based media for transporting. Studies show that most viruses are sufficiently stable to withstand a transport time of 1 to 3 days when it is in appropriate transport media. The specimen should never be exposed to extremes in the environment. So if you're in an extremely hot climate, you will need to refrigerate your specimen. Modern viral transport media allows for effective use of viral cultures and culture enhancement techniques for the diagnosis of viral infections.
Hi, my name is Kimone, and the topic that I have is which possible laboratory tests using the sample can you do to confirm that this is a viral infection? Such tests include virus isolation, immunohistochemistry, antibody detection, reverse transcriptase polymerase chain reactions.
Hi, I'm Sarvesh. The question was, which is the most likely virus to cause diarrhea in feed-out cattle? The virus that causes BVDV is Flaviviridae pestivirus, which is the most widespread cattle pathogen. Flaviviridae has monopartite, linear single-stranded RNA genomes of positive polarity. There are two biotypes of Flaviviridae pestivirus, non-cytopathic and cytopathic. The non-cytopathic biotype BVDV will establish a persistent infection in a fetus when infecting a dam in early gestation, and these cows will then be infected for the rest of their lives, causing them to shed the BVDV virus.
Hi, my name is Taalia. My question was, can this virus easily grow in cell cultures? The virus can grow in cell cultures, but can have high probabilities of becoming contaminated and non-viable. Infection of the virus in utero can lead to virus and antibody contamination. All serum samples need to be screened to ensure they are uncontaminated.
Mamphokoane. My question was, many bacteria can also cause diarrhea in cattle. How will you determine this is not a bacteria causing the disease? The diarrhea in cattle caused by bacteria infection is usually a yellow and tan color and smelling very bad. There may be bits of mucus and blood in the stool. These animals are usually depressed and may not have a fever. The only way to confidently diagnose is through bacterial culture. In bovine viral diarrhea, clinical signs are highly variable. Signs of acute infection include fever, lethargy, loss of appetite, ocular discharge, nasal discharge, and oral lesion. BVDV infection almost always produces some degree of immunosuppression determined by virulence of the infecting strain. Infected cattle shed BVDV in various body fluids and tissue. Bovine viral diarrhea includes several distinct diseases and syndromes like respiratory infection, thrombocytopenia syndrome, persistent infection.
Hello, my name is Zandre, and my question was, how will you be able to confirm the morphology of the virus, and which morphology can this virus possibly have? The first technique is antibody assays. An acute infection can be confirmed by the presence of a specific IgM. antibody assays are tested through the enzyme-linked immunoassay technique, otherwise known as the ELISA technique. The second technique is the direct examination. Electron microscopes can be used to visualize viruses, marking virus-infected cells by labeled antibodies. The antibody binds to its corresponding antigen. The cells can then be detected under the microscope. The third technique is culture. Viruses only replicate in living cells. Replicating viruses change the appearance of the cells to induce a cytopathic effect. Different viruses cause different types of cytopathic effects. The fourth technique is molecular technique. Polymerase chain reaction can be used to detect viral and mutant viruses. It can also be used to detect any DNA sequence, and therefore it is a viable option to test for specific viruses. The bovine viral diarrhea virus is a near-spherical viral virus which displays a capsid surrounded by a phospholipid bilayer and no visible spikes.
Hello, my name is Willem. My question was, explain the specific eclipse phase of the replication of this virus. The eclipse phase is the period of time between the infection by the virus and the appearance of the mature virus in that cell. Bovine viral diarrhea virus is a positive sense RNA virus. This RNA can function as a genome and a messenger RNA and can be directly translated to proteins in the host cell by the ribosomes in the cytoplasm. During the eclipse phase, the RNA genome replicates to produce new variants. In this period, the viral infectivity cannot be recovered, and this phase will last till new infectious variants are made. The eclipse phase of the bovine viral diarrhea virus is approximately 8 to 10 hours.
Hi, it's Taalia again. This question was, is this virus prone to mutations, and what are the possible types of mutations that can occur in this virus? The bovine viral diarrhea virus is an RNA virus. Mutation and variation occurs, but mutations are not as common as variations. Various types include genetic, antigenetic, and pathogenic variation. Conclusively, there are two genotypes known as BVDV1 and BVDV2. Both separate species fall under the pestivirus genus.
Hi, my name is Kimone, and my topic was, how does this virus enter the host, and how is this virus shed into the environment? Bovine viral diarrhea virus is a pathogen associated with gastrointestinal, respiratory, and reproductive diseases of cattle worldwide. The virus can cross the placenta during early pregnancy and result in birth of persistently infected calves, which are capable of shedding large quantities of the virus throughout their lives and are considered the primary reservoirs. The main transmission route in the infected herds is direct contact with a persistently infected animal. Other methods include horizontal transmission, which is contact with an infected virus-shedding animal, which may be direct or indirect via inhalation or ingestion of the virus-contaminated particles. Common mechanisms of horizontal transmission include fomites, which are feed, water, and equipment such as nose tongs, milk bottles, and needles. Others include palpitations, secretions and excretions, and crowding of animals with a respiratory type of the virus, as well as vectors such as horseflies, stable flies, and herd flies. There's also other methods which are transplacental infection, which is from an infected mother resulting in fetal death, malformations, acute syndromes of the neonate, and immune tolerance and lifelong viral persistence. There's also vertical transmission, which includes contaminated semen, embryo transfer, and contaminated modified live vaccines. Persistently infected animals constantly shed large amounts of virus throughout their lives in the environment where it is in contact with non-infected animals and act as a source of infection. The virus is thus presented in saliva, nasal discharge, milk, urine, feces, and semen. Also, flies, gloves, and nose grips can also cause a spread in the disease. Thus, close contact between cattle increase the risk of danger.
Hello, this is Pamela again. My question is, being an intracellular pathogen, which mechanism can be used to evade the immune system of the host? There are various ways of evading or subverting the immune system. Example, non-cytocidal infection, cell-to-cell spread, infection of non-permissive, resting, or undifferentiated cells, restricted gene expression, evasion of neutralizing antibodies, induction of immune tolerance, and immunology privileged sites.
Thanks for watching our video.